Our next presenter is Dr. Cristina Guarducci of the Dana-Farber Cancer Institute in Boston, who will present GS307, Clonal Evolution and Mechanisms of Acquired Resistance to CDK4/6 Inhibitors in ER wild-type and ER mutant breast cancer.
So thank you for the opportunity to present today. And on behalf of my colleague, I'm going to show you the results of the study, Clonal Evolution and Mechanism of Acquired Resistance to CDK4/6 Inhibitor in ER wild-type and ER mutant breast cancer. Oops. So we all know that multiple mechanisms of acquired resistance to CDK4/6 inhibitor have been identified in clinical and preclinical studies. However, it remains unknown if these mechanisms are pre-existing or acquired during treatment. Also, there is evidence that ESR1 mutation have an impact on the outcomes and acquisition of resistance to endocrine therapy and palbociclib. However, the role of the ESR1 mutation in the resistance to the CDK4/6 inhibitor component of the endocrine therapy and CDK4/6 inhibitor combination is unknown. Clinical and preclinical evidence also suggests that there may be differences among the available CDK4/6 inhibitor, and it's still not clear if the mechanism of resistance to the different CDK4/6 inhibitor are disparate. So based on this, we decided to perform a study to try to answer to the following question. So first of all, is the acquired resistance to CDK4/6 inhibitor due to the selection of pre-existing resistant clones or to the acquisition of resistant alteration during the treatment with CDK4/6 inhibitor? And the answer to this question might have clinical implication because if the resistance of clones are pre-existing, then it would be important to treat patients with therapeutic strategies targeting the resistance of clones upfront. Other question we had are, second question, do the ESR1 mutation impact the clonal dynamics and the mechanism of acquired resistance to CDK4/6 inhibitor? And also, are there different clonal dynamics and mechanism of resistance to abemaciclib versus palbociclib in ESR1 wild-type and ESR mutant tumor? So to answer this question, we perform an in vitro study engineering the cells with a DNA barcoding library, the clone tracer library. So what is a barcode? A barcode is a DNA sequence that gets integrated into the genome of the cells and is passed to all the daughter cells. So by tagging a cell with a barcode, is it possible to track, to follow the clones within a cellular population? And in this absence of a selective pressure, we might expect by sequencing the barcodes to find that the barcodes are equally distributed within the cellular population. While in the presence of a selective pressure, some clones might have a growing advantage, and therefore by barcode sequencing, we might expect to find that some barcodes are enriched in the cellular population. So to study the resistance to palbociclib and abemaciclib in the presence or absence of the estrogen receptor mutation Y537S, we DNA barcoded doxycycline-inducible ESR1 mutant MCS7 cells. So after we barcoded these cells, the parental cells, were divided into two groups, the cells treated with doxycycline and the one untreated, because these are cells that, if treated with doxycycline, express the Y527S estrogen receptor mutant on top of the wild-type endogenous estrogen receptor. So this population of cells, the wild-type and the mutant, were then further divided to receive increasing concentration from 50 nanomolar to 1 micromolar of palbociclib or abemaciclib, or the vehicle of the drug, so the controls. By barcode sequencing, we check the distribution of the barcodes at different time points, an early, a middle, and a late time point. And this is also the time point that corresponds to the time of resistance to palbociclib or abemaciclib. At this time point, the cells were also subjected by whole exome sequencing and RNA sequencing in order to study the genomic and transcriptomic profile of the cells. And importantly, all the experiments were performed in triplicates. And this is a key point in our study because it allowed us to understand if the resistance is pre-existing or not. Indeed, if the resistance is pre-existing, then we expect by barcode sequencing to find that the biological triplicate are enriched with the same barcodes. While if the resistance is emerging during the treatment, then we expect by barcode sequencing to find that the different biological triplicates are enriched by different barcodes. So first of all, we look into the evolution of resistance to palbociclib. And here in this heat map, you can see in blue and in yellow the palbo-resistant model and the respective control in the annotation bar of the heat map. So in this heat map, each row is a barcode, and the color represents the level of enrichment of each barcode. And specifically here, we show the barcode with an enrichment with a fraction above 0.01 in at least one condition. So when we look into the palbo-resistant model, we found that the biological triplicate clustered together and shared the same enriched barcode, indicating that resistance to palbociclib is the result of selection of pre-existing subclones. Also, the first splitting of the barcodes was based on the expression of the SR1 mutation, and the SR1 mutant palbo-resistant cell, highlighted here in the pink box, displayed distinct clonal selection compared to the SR1 wild-type palbo-resistant cell, highlighted here in the light blue box, suggesting that the Y537S SR1 mutation impacts the selection of the palbo-resistant subclones. We performed the same analysis in the abema-resistant model and the respective control, indicated here in the annotation bar in green and yellow, respectively. And here we found, again, that the biological triplicates clustered together, suggesting that also the resistance to abemaciclib is the result of selection of pre-existing subclone. But in this case, the first splitting of the barcodes was based on the resistance to abemaciclib, and the SR1 wild-type and SR mutant cell had a high number of overlapping selected clones. When we directly compare the palbo-resistant models and the abema-resistant model, we found that the palbo-resistant and abema-resistant cells are different, and the difference between the palbo-resistant and abema-resistant cell is more pronounced in the SR1 wild-type setting compared to the SR1 mutant. So next we look at the clonal dynamics of the resistance to palbociclib and abemaciclib. And in these fish plots, each color represents a cellular population. So when we compare in the palbo-resistant model the mutant on the bottom versus the wild type on the top, we found that the SR1 mutation led to an earlier and more heterogeneous clonal selection. When we look into the abema-resistant model, again comparing the mutant versus the wild-type cells, we found that the SR1 mutation led to an earlier clonal selection, but didn't have a strong impact on the clonal diversity. Functionally, we found that the palbo-resistant model retained sensitivity to abemaciclib, while the abema-resistant cells were cross-resistant to palbo. Genetically, we found that the barcode clustering mirrored the genetic clustering based on the presence-absence of mutation detected by whole exon sequencing, suggesting that the selected clones represent genetically distinct cellular subpopulation. By single-cell RNA-seq, we found that the abema in palbo-resistant model display a distinct transcriptional profile compared to the naive cells. Also, the wild-type resistant cells were transcriptionally distinct compared to the mutant-resistant cells. And the abema-resistant model had a different transcriptional profile compared to the palbo-resistant cells. When we look into each model, we found that each model consists of transcriptionally heterogeneous cellular population. And overall, by bulk RNA-seq, when we look at the top differentially expressed genes across the different conditions, we found that the SR1 wild-type abema-resistant cells show overexpression on interferon response-related genes. The SR1 wild-type palbo-resistant cell show overexpression of EMT-related genes. The SR1 mutant, palbo, and abema-resistant model were characterized, not surprisingly, by the overexpression of estrogen response-related genes, and also they show also increased overexpression of interleukin-2 STAT5 signaling. And the EM mutant, abema-resistant model were characterized by overexpression of KRAS signaling-related genes. So, in conclusion, with this study, we found that resistance to CDK4/6 inhibitor is likely due to the expansion of pre-existing resistant clone, suggesting that targeting resistance upfront could delay the acquisition of clinical resistance. Also, although the SR1 mutant cells are sensitive to CDK4/6 inhibitor, the Y537S ESR1 mutation shape the clonal diversity and dynamics of the acquisition of resistance to CDK4/6 inhibitor, more so to palbociclib and to a lesser extent to abemaciclib. And the clonal selection and transcriptional changes during the acquisition of resistance to palbociclib and abemaciclib are different, especially in the SR1 wild-type setting, highlighting the difference between the two drugs. And with this, I really would like to thank my PI, Renat Jeselson, and also the member of the Jeselson Lab, in particular, Agostina Nardone, Douglas Russo, Avery Faith. And also let me thank Miles Brown and all the Brown Lab member, and Francisca Michor and Simona Cristea for their support and contribution to this work. And with this, thank you.
Thank you, Dr. Guarducci, for that fantastic presentation. We'll now take questions from the audience, and we'll start at microphone 3.
Thank you. That was very interesting. Nick Murray from Adelaide. I'm interested in the reproducibility of your results. Have you done this experiment multiple times? And if so, do you see the same patterns emerging each time?
So we haven't repeated the experiment in other cell lines. So this is just the model that we have developed. And we performed the experiment in biological triplicates. So it's like independent experiment. But we haven't done the experiment in another a newly barcoded population or in another cell line.
Microphone 4.
Hi, very good talk. I'm Mark Hafner from Genentech. I do have a question, and maybe I missed that. But did you look into the transcriptional state in the initial condition before treatment, and if the barcodes between the two treatments were aligned or not?
So yeah, of course, we also had the transcriptional profile of the parental cells or the controls. Here we just show the direct comparison or the differences among the condition because we thought that it was more interesting. But of course, because of lack of time, we couldn't show everything. But yes, of course, we had them, and there are differences, of course, compared to the controls. So I don't know if you have a question specifically about this.
Yeah, in terms of the clone selection between the two treatments, if they were coming, like you have multiple clones, were they actually the same state?
Yeah, so you mean the comparison between also the triplicates? Yeah, so actually that is something that we have not done, so we should do, of course. We do not expect to find big differences, huge difference, considering that the replicates, at least by barcode and also genetically, are similar. But of course, it might be that we can find some differences, maybe due to, mechanism of adaptation.
Thank you.
You're welcome.
Dr. Guarducci, if you were asked, based on the results of your analysis, to help to design a clinical trial to see if the addition of another agent to the combination of endocrine therapy with either palbociclib or abemaciclib, what type of agent might you suggest?
Yeah, I think it's hard to say because of this heterogeneity. So I think that for sure we need more preclinical studies and maybe also analysis in patients to try to understand this heterogeneity, not only at the time of resistance, but also at the beginning before treatment, to try to understand what could be the best combination, because I think that it's a challenging question.
Thank you.
Thank you, Dr. Guarducci, for your
Thank you.
presentation.